Hello, everyone. In the previous lectures, we discussed that how the gene expression can be identified in the different TCGA datasets, and we use GPR software for that. And this GPR software actually contains a lot of different tools and methods to visualize the gene expression. The title is the Gene Expression Profiling Interactive Analysis. So this is an interactive portal where you can see the pattern of gene expression across tumor versus normal, and you can also see the how the gene expression associated with survival. This type of plots we have discussed in the previous, and we also discussed how they actually what does it mean, like what is the blue line, what is the red line, what is x-axis, what is y-axis, what is p-value, and so on and so forth. Also, now in this tutorial, what we will see here is that what is the violin plot, how the violin plots are generated for a particular gene, and what are these box plots, how we can see these box plots here. So basically, uh, what we will do is that we will jump to these different features which are mentioned here, like profile. We have seen the profile, then the box plot, then the stage plot, then survival we have seen. So if we click the box plot, we will go into this their portal, where we can have the options that, for instance, we want to look for any specific gene. In the previous lectures, we saw the pattern for FGFR4, so we will again select here pattern FGFR4. So, and the log fold change is 1 is the cutoff. The fold change 0 um, is actually log fold 1 means that there's a two-fold change, and p-value cutoff is 0.01, which is stronger than 0.05. Usually we take 0.05, but if you want to make it more stringent, we take 0.01. And these are now the different cancer types, their abbreviations. And for instance, if we select any of these, for example, lung adenocarcinoma, and we add that into this feature, and then we want to scale by log, yes, because it will normalize the values, the TPM values. And then we want to match TCG normal and GTEx data, and yeah, you just have to keep these features default, and then click plot. Now, if you know that this is a large scale whole transcriptomic data, but by just going into this website and writing the gene name, you can get the publication quality images, p values, and all the details here. Now, this is the plot, this is the box plot. You can see that there are two boxes here. And what is a box plot? Box plot basically is the this red and gray. You can see two boxes. If I speak very simply, in a very simplistic way, that actually here there are there are multiple, you can say, bars here. For example, if we take this one only as a box plot, and we see that this is the line here, this is a line here, then there's a box between it, and there are a lot of dots values between these. So these dots are actually the expression values of in each sample of FGFR4. Same for this gray one. These all dots are the expression values in the particular sample. Then this is the maximum horizontal line here, and there is a minimum horizontal line here. And these are some values which are outside this maximum and minimum. So what this each line means, this line which is at the bottom, this one for example, this one in a box plot, these are actually, this is the minimum value in a in normal distribution. Here gray is the normal. If you see here, gray is the normal and red is the tumor. If we talk about the gray here, this line, this horizontal line, if you can see my mouse, this is the minimum value in a in a distribution of normal samples. And this one which is at the top is the maximum value in normal samples. And between all these are actually the dots, which dots are the values which are distributed between the maximum value of a sample and a minimum value of a sample. And then these are some, these, this is the bar, This is actually the third quartile. The third quartile is actually the 75% of value. So till this point, in the normal samples, the 75% values, like from the 0 to 75 maximum values, are distributed from this line to this line. And then this is the last box. This one is the first quartile, which means that the 25% values are below this one. These are all the 25% values, and these are all the 75% values, from starting from here to here. And from here to here, these are 25% values. And this bar, the horizontal bar you see in the middle, is actually the median. Median is, what is the median? Like, if you divide the sample to two groups, it's a very simple concept. Like, if there are the middle value, you can say, of all the samples is the median. And then these values which are outside this bar, these dots are the outliers. These ones. And so now, if you see, again, for the red one, it means the same, that this is maximum, this is minimum, this is third quartile, this is first quartile, this is the median. So you can see a very strong difference between the expression pattern between the tumor samples, which are red, and normal samples, which are black. So there is the median value of both is strongly different. The third quartile value is strongly different. So you can, if you consider any of that feature, it means that the FGFR4 expression is significantly different between tumor and normal samples. And normals, it is very high. And this steric actually means that the significance, if there is a significance, usually the different statistical tools take it as a statistically significant value. So they just put star, like here the statistic 0.01 is significant, so steric means that the value is less than 0.01. So I think I thought to explain the box plot, then the volcano plot, but for now I think it's enough. In the next video, I will explain what is the volcano plot and how you can do volcano plot analysis for a single gene and maybe for the multiple genes as well. Thank you. Bye bye.